DNA collected from seawater may solve mysteries about world's largest fish. Using only the DNA from sloughed-off cells floating in the ocean, scientists have been able to determine the population size and genetic properties of one of the world's largest and most mysterious animals, the whale shark. The work marks the first time researchers have been able to use so-called environmental DNA, eDNA, to estimate the genetic characteristics of an aquatic species, and it could help scientists study the population and health of a wide range of marine animals without ever setting foot in the water. The results are a conceptual advance, says marine biologist Ryan Kelly of the University of Washington, UW, in Seattle, who was not involved with the research. They push the boundaries of what is possible to do with environmental DNA. The research traces its origins to one summer day in 2007, when a worker on a Maersk oil platform in the Al Shaheen oil field off the coast of Qatar saw a surprising sight, a group of roughly 100 whale sharks feeding near the surface. Scientists hadn't realized that the fish, the world's largest at roughly the size of a school bus, frequented these waters, and the gas field soon became a hotbed for studying this endangered species. Whale sharks can be difficult to locate, however, because they are often far out at sea. Those at the Al Shaheen oil field were more than 80 kilometers from the coast in the Arabian Gulf. Biologist Eva Egling Sigsgaard at the Natural History Museum of Denmark at the University of Copenhagen and her team collected seawater containing skin cells along with cells from urine and feces naturally shed by the whale sharks and other animals. The researchers isolated the cells, extracted and sequenced the DNA within them, and then used software to assign some of the D. The researchers then used the DNA to estimate the number of reproductive female whale sharks, roughly 71,000. Whale sharks appear to be genetically split between two groups, and this estimate reflects the female population of whale sharks in the Indo-Pacific Ocean group. This number was broadly consistent with estimates from actual tissue samples, the team reports online today in Nature, Ecology and Evolution. In a related study published this month in PLOS One, scientists showed that eDNA collected off Greenland revealed which fish were most likely to be caught by deep-water trawling, a finding that could revolutionize how marine species are studied. That's because using eDNA is a cheaper, easier option than dragging nets across the ocean.